Okay, thank you very much. Thank you to the organizers, thank you to the chairs to invite me here to be today, to tell what we do, and these very interesting and exciting days, talking about many things that are related. I want to thank to the previous speaker and to the chair to show most of the things that I'm going to tell you, that I'm happy. We have maybe more time for discussion about other things. And now I would like to introduce my institute first. We are an institute not related with food safety, not related with food technology. We are with food and health. Our aim is to study the relationships between nutrients and diet with non-communicable disease from a molecular perspective point of view. You will see that our pillars are different groups that are related with precision nutrition and aging, something that was discussed before, cancer, obesity, cardiometabolic disease, and childhood nutrition. And then we have a clinical trial platform that is genial, that is fully equipped to do many studies from the biomarkers, laboratory, body compositional for the people that come in. We have around 3,000 volunteers inside. And then we have platform for organoids and organ on a chip to do in vivo assays with all the computational stuff for omics data. No? The idea of this platform is to provide personalized drug combination and products to promote health, no? And this is right at our scope. I hope that we can collaborate with different ILSI and all of you that are interested on us. No? Well, I'm going to start about human microbiome. I will focus on this topic that the chair asked me. You know, here there are different databases that have been created a lot of data in the last years. I think that who is not interested now in microbiome? I think that is a hot topic now in the area, not only for the microbiome itself, but for probiotics, prebiotics, and for food. You can see that there are many examples, but in fact, we have very far away compared with what have been done previously, for example, in transcriptomics or in epigenomics, in cancer and in other areas. We're a little bit far away. But we can start to apply artificial intelligence tools. No? Here, this was presented before. I will not go inside because you know a clear idea about this. Artificial intelligence can be about speech recognition, natural language processing, others, but here in microbiome area, we are using machine learning techniques, no? To predict or to, as was said before, identify patterns that we can predict different outcomes. Previously was very well stated, the different groups that we can use, supervised, unsupervised, semi-supervised, and others. And basically, they are going to be used for feature generation to create new variables that are going to be studied. In food, it's going to be to study food diet response, disease severity, including some food allergy or others, for data integration, comparative microbiome analysis, food prediction to identify biomarkers that can be used for gene prediction. This is a bit a few of how it's being used. No? Okay, my group is part of the machine learning for microbiome cost action in Europe. This is a group of around 150 researchers across all Europe, all institutes. It's multidisciplinary. We have microbiologists, clinicians, bioinformaticians, statisticians, engineers that try to see how the machine learning techniques are being used in the microbiome area, in the human microbiome area. You can see here, this is a revision that we published a time ago, about 250 papers that we could find. Most of them is about what is the microbiome composition of different organs or different tissues. Then we have diet, of course, is really important. And then we have other diseases that are related with food, like could be obesity, diabetes, inflammatory bowel disease. No? That is the main focus now that the people is looking at in this kind of studies. The steps to prepare the model building, this is not so simple, like clicking the computer and you have the outcome. You have pre-processing steps with normalization, filtering, batch correction, feature selection, select which are the variables that are going to be important to build the model. We have different steps, filtering, wrapper, embedded methods, and then the modeling. No? You use the model and you need how to use predict algorithms, estimation, performance optimizations. For all of this, we don't have benchmarking yet for human microbiome. This is something that we combine here in the microbiome cost actions. This is something that an effort that is really needed. How we are going to address each of these questions and how is the most proper method to do this. And we identify many challenges about the application of machine learning in microbiome. Some of them are related with machine learning itself, handling outliers, non-deformative features, but others are really highly related with microbiome. No? Some of them, okay, when we use 16X or some metagenomics, we have different type of sequences that we have to work with them. We have sparsity and variability. We have long matrices with many zeros. Many of them, they don't follow a Gaussian model. And they are not normal, that are normal. I mean that Usually many methods that we used in the past we cannot use. There is intervariable dependency. You have many microbiomes that are going to cooperate or they are going to be together. That means that they concurrence. Data dimensionality. In general, although I show you this table with big numbers of samples, in general, most of the study have about dozens or hundreds of samples. They are very limited. That means that we have more variables than samples that we have, and that makes a complexity in how we are going to approach the analysis of these kind of things. No? And then we have another one that is specific, more specific for microbiome, that is compositional data. No? Even though when you include one new feature, you need to reduce other features. I mean, that means that gives a problem because the balance between the microbiome that you can see between two different samples can be equal, although as you are thinking in relative numbers, they can be, you see in the numbers, are different. Okay? And this gives a complex problem. I will not go inside of all of them because they will need a specific section. I will just go to the compositional data. Inside of the co-section, there was a group that reviewed how the people is doing this kind of things of transformation of the data. You can see here that 30% of the papers that they review didn't say nothing about how they transform the data. This is go to the open access and reproducibility and all these kind of things that we have discussed previously. This is a big problem for the community. Then we have another big problem, number of DERs that they use relative abundance, set score, log transformation that was very used in the past, well, in the past and now in the transcriptomics, for example, area, that when you are working with absolute numbers. And there are little ones that are using specific transformation methods for compositional data, that is the microbiome data. Of course, that you can, in the years you can see that this is starting to increase in the last year, but it's not like in the others method. No? That means that it can have some bias and have problems of interpretability about the result that we are getting in. No? Well, this is just to point it out that we have problems that we have to solve first before go to very specific question on other questions. Then you can see that although we have these problems, the interest of the community is increasing. And you can see in the last year, 2021, 22, the development of software, specific software of machine learning software for human microbiome having increased a lot. Here you have a revision with many methods that have been developed for sequencing, I have to say, most of them. You can see that most of them are centered in feature generation, while other areas are poor in the development of methods like data integration. There are others. I invite you to see the paper, really, to go there if you are really interested on that. But what we can see is that the development of software is being high in the last years, no? And it's going to expect that it's going to increase in the next following years. But we don't know, when you are a bioinformatician or analyst, you don't know what software you should use. Because we don't have yet benchmarking approaches to try to see how is the difference between the different algorithms, the different software that we can really work on it. No? Well, I just invite you to work in the, or to check the different papers that the community of co-section have been published in collaboration with Frontiers in Microbiology. Other works are coming in the next year. And I think that these are very helpful resources that you can check about this, the problem, the challenge, how the people is trying to solve, and many other things. Then I'm going to talk about a couple of examples that we do in my group about how we manage the microbiome, taking into account these problems. No? We have one study in celiac disease. You know that celiac disease is an autoimmune disease characterized by gluten intolerance. It's around 1% in the world. It's genetically susceptible, and you have gastrointestinal and extraintestinal symptoms. The unique treatment that is known from the 60s of the last century is the gluten-free diet. I mean, you can see this is innovation. I mean, from the 60s, we don't have any other resources to treat the gluten-free diet. Okay, we started to collaborate or try to work and recopilate all the work that we can find for sequencing for microbiome data. This is why this is important, the open science, because when you have maybe new ideas, you can bring other data and try to interrogate that data to solve a problem or try to interrogate a problem. We are high supporter of open science. You will see that all is published in open science. You can access. We identify many different studies, thanks to the authors, from the upper track and lower track. In the Einstein, we identified some different biomarkers, but what I tell you is, okay, we went to create a machine learning model to try to classify patients with celiac disease with non-celiac disease. We were really able to get a good results with a random forest, this one method. I will not go into the details. And we can really identify pretty well those patients that are following a gluten-free diet, but we are able to identify too those patients that are diagnosed with celiac disease, but they are not following a gluten-free diet. This is important because, okay, these are not invasive samples. Now the diagnosis of the celiac disease is with a biopsy. I mean, you will see a lot of people that say, okay, the gluten for me is not good for this food and dietary, but the diagnosis today is by a biopsy. I mean, this is important to take in account. We can identify those markers, and we have a list of these markers that could be important, and we are able to identify both in both cases or not. This is one way that most of the people is using machine learning for classifiers. Classify food with microbiome, classify diseases related with food or other things. We wanted to go a little bit deep on this, and we know that there is a condition that is the non-responding celiac disease. There's people that after 1 year following a restrictive diet, gluten-free diet, they have yet some symptoms, extra gastrointestinal symptoms. We know that this is around 30% of patients. And this is a very poor condition. I mean that if the people have celiac disease, they are matching this gluten-free diet, they can have yet problems, and they don't have solutions yet. As I say, there is no other treatment, there is no other things. No? I mean, for us, it was really important, and we thought that the microbiome could have some issue here, something key aspect to do. We collect a few patients, 40, I have to say, but we try to do a very deep phenotyping of them from the clinical point of view, inflammatory markers, markers for mucosal integrity, different questionnaires about many things, and then we have sequencing data and metabolomic data of the stool samples. No? We started working with this, and we apply here machine learning methods. Anyway, we try to identify which are the variables that vary between this cohort highly. We identified 43 about 99 that we assigned, and we were able to identify 2 groups. I mean, this is 2 groups that we are really able to characterize. We know that these groups, they have good adherence to the diet by questionnaires and by all the biomarkers that are not here, but we know. But then they have high difference in how are the symptoms that they are getting after adherence of this diet. No? We already see this. Okay, this is really interesting. Now, let's see what is the role of the microbiome that we have here with the data that we have. We went to the alpha index, different alpha index. We didn't see really nothing. Of course, they are celiac disease. They could have both dysbiosis. That is okay. Then we went to the abundance. We did many different things, supervised and unsupervised, and we couldn't see really differences. No? Okay, here the artificial intelligence and the machine learning didn't help a lot. We can say, okay, maybe there are few samples, that's true. Maybe the methods that we are using are not the most appropriate to do. Maybe, can be. We really don't, but this is what we have. And there are many projects and many studies have this kind of numbers, 50, 100, something like that. And maybe we need to shape our mind how we think, how we approach this, no? How we check this. No? And we start to think in networks. Why we think in networks? Because at the end, the microbiome is a community. It's an ecological community. And they communicate, they share genes, they share functions, they do this kind of thing. No? And we found this paper that explains, okay, you can create a co-occurrence network. That means that with the patients that you have, you can see if they co-occurrence in the same patients or not in the different clusters. No? These are the dots that we have here. If they are bigger or not, this depends on the abundance. And then we have edges that can be increased if they are correlated, if they co-occurrence, or they are in red if they are not co-occurrence. They never are together in the same patients on the same cluster. No? Something like that. And we say, okay, let's go to apply this kind of techniques because maybe this give us more information. We do that, and you can see here the 2 clusters that we can identify in the different clusters, cluster 1 and 2, and here we can identify some keystone taxa. This means that they are important to the stability of the network. That means that these species should do something inside, they put in common other microorganisms, and they give stability to the network. We identify these keystone taxa, different ones for them. This is nothing about the abundance, that is the usual approach that we do. If a microbiome is there in the sample and not in the other, this is a little bit different. And we identify some of them. Then we compare them. We create another network to compare what are the differences. And here you can see, for example, that we have one of them, Roseburia hominis, that is related with extended gut barrier function. This is high correlated in the cluster 1 with other bacteria. Good. And then we have another one that is Ruminococcus torques, that is high related in the cluster 2, that is with disruption of the gut function. We started to identify biomarkers that with the eye we couldn't in the previous lead that could be interested for this and caveats. And then you can see here that you have Bacteroides silaninus lovens. I mean, in both of them, related with both of them. That means that, okay, sometimes we are going to have microorganisms that are going to be in both places, but can play a different role depending on how is the relationship with the other bacteria, okay? That means that this could be help to us to understand this, no? Then we went to the metabolome, with the metabolics. We collaborate with this with Alejandro Cifuentes. I think that some of you know him. He is close in the street. And we went to them and they identified 77 metabolites. But it was a little bit worried because only 3 was from the microbiome. We say, okay, here there is a problem. And what we did is, okay, we have the sequence. We are going to use different methods that can predict from the sequence what is the metabolite that should be there. No? We do that, and we identify that we should find 80 metabolites for the microbiome. We back to Alejandro. We discuss about this. They review the data that we have, and at the end, they come with around 372 metabolites, 40, almost 50 of them related with microbiome. No? That means that here the AI helped us really to increase and to moderate this. Maybe Professor Wizard that is here, maybe is more expert than me in metabolites and have their other methods, other ways really to find this better, but this is the way that we are using now. We take the sequence, we predict, and then we go to the metabolomic people to try to identify with what is there. No? And here we create a more complex network. Here you can see here you have these pieces in cycle. The diamonds are different metabolites that we have identified, and then in a square we have the functions. That means that we can relate how is the abundance of the species of the metabolites with the function. Because at the end the idea is not to have, for us, is not to have a list of species that we don't know what is the function. At the end the important is the function. And we know that although some species can be related at the phylogenetic tree, for the function could be really very different. And this is what we think that is really important. And this is why we think that combined IE with networks is really interesting to this. No? Here we identify one species that you can see here that is increasing the different metabolites that we have in our experiment, and this is related with different pathways. No? At the end, as a summary, going a little bit in the biology, we see that the cluster GAN have better markers of gut barrier function, nitrogen and amino acid biosynthesis, that is for good function of the gut. And then we see that there are inflammatory markers in the second cluster, activation on immune response and protein degradation. I cannot go beyond of this because we don't understand yet completely how this works, because you see we have this complex. In fact, we have asked a 4 million project to the European Union to go on deep on this. And we are trying to identify what happened with this, no? and with this type of things. Just to finish in a couple of seconds, I would like to tell you the experience that we have. We have a community in Elixir. Elixir in Europe is the data science platform. And there are many communities for many things, microbiome, metabolites, and others, but we have one for food and nutrition community. Of course, we were very ambitious at the beginning, and we wanted to define individual health status, for develop individual advice, and at the end, try to understand the complex dietary and food intake biomarkers. No? This is a high question that we want to solve. No? At the end, we really realized that we have many problems, as we discussed before. As standardization and interoperability, we need to develop more ontologies, how we are going to do the standardization of the questionnaires, tools for interoperability solutions, and others. No? Data availability, we back to the open science, is really important, as we saw, to have this with a lot of metadata as much as possible to develop services. No? We started about microbiome because it's a topic, and the first question was where we put our data, microbiome, sequencing, metabolome, and phenotypic data. No? And this is what the first where we are starting. We have identified some platforms in Europe, Phenotype the base for data about food, metabolites for metabolome, and Magnify, but then comes another problem. How we combine in the same studio if we have one here, another here, another here. No? And we are working now to automatize tools that can bring together the same studio, different studios that you can work easily and fast. You know? I mean, this is something that we are developing, and you are interested, and just, then I finish. slide for the take-home message. Many challenges we have to address in AI applications yet for microbiome data. We need benchmarking efforts. Microbiome is complex and requires integrate different approaches, AI and networks, as I showed you. Networks can be a good solution for small data sets. More collaborative efforts are required to understand microbiome complexity. You know, we have for cancer, we have the Alliance of Cancer Genomics, we have the PNomics, but we don't have an alliance yet for microbiome, even in the food or not. Be fair with your data, take advantage of the resources, tools and software from the community. And the last one is check all the great resources and paper from machine learning for microbiome that we have been published. Thank you very much. This is all the supporters of my group, different fundings. This is the people that collaborate with us in some of these projects. Well, I would like to thank all of you for your attention. I hope that I'm sorry that I cannot answer what is your good microbiome, but you know what are the challenges, pitfalls, and I hope that we can collaborate with ILSI and with other institutions that are here to answer those important questions. Thank you very much. Thank you very much for your attention.